Hello everyone. Today we're continuing our deep dive of Richard Dawkins and Yan Wong's book, The Ancestor's Tale. In this episode, we're going to discuss why no modern organism is our direct ancestor, and a whole lot more. So let's jump right in. Here in episode 40, we are leaving the vertebrates behind. The last tale left us at the last common ancestor of all vertebrates around 525 million years ago in the early Cambrian, a period known for its extensive radiation of bilaterians. We've discussed the evolution of life in the Cambrian ad nausea on this channel, so we're going to refer you to the videos we did collaboratively with Professor Dave for the deep dive on it. The short version is that the Cambrian, which lasted from 541 to 485 million years ago, saw multi-tiered radiations of animals. The first half of the Cambrian saw the radiation of stem non-bilaterians and stem bilaterians, both of which originated in the preceding Ediacaran period. More on them in the next few tales. While the second half witnessed the radiation of crown members of both groups. And as we mentioned in the last tale, these diversification events continued into the following period, the Ordovician, in which we met the first jawed vertebrates. The potential reasons for the Cambrian diversification event are myriad. Rises in oceanic oxygen levels, relaxing animal size constraints, the advent of the ability to burrow in sediment, the origin of biomineralized parts triggering an evolutionary arms race, the origin of camera eyes, as well as others. Or, more likely, a combination of all of the above. Our world is not the result of monocausism, so what was around then? While early stem vertebrates had camera eyes, they lacked mineralized hard parts and reached just a few centimeters in length. These include Hycoichthyes and Meliconmingia from China, and Metaspergina from British Columbia, Canada. As one recent paper notes, quote, by combining Meliconmingids and Metaspergina data, a better reconstruction of the most likely Cambrian vertebrates is possible. A jawless fish with a pair of large anterodorsally facing camera eyes, a small median olfactory organ, 5 to 7 pairs of gill arches, a stomach, a series of chevron-shaped myomeres, and a median fin web. Close quote. These stem vertebrates look like little more than worms with dorsal fins, and at this point, that's what we should expect. This is what basically anything that resembled a vertebrate looked like during the early Cambrian period. We once again run into the common theme that we see in almost every episode. While modern groups have many traits unique to them, such that we can easily tell the differences between them, as we go further back in time, we will pass beyond points when those distinguishing traits have evolved. This is why the further back we go, the more similar related groups will appear until it's difficult to tell them apart. Regarding vertebrates, we've left behind hair, amniotic eggs, limbs, lungs, jaws, scales, and bones, and even skeletons. The characteristics we can use to identify animals as stem vertebrates are rather few in number. For this reason, a variety of Cambrian worm-like animals have been variously identified as stem vertebrates, stem chordates, or even stem deuterostomes. Before we get into fossils, a brief reminder is in order. Deuterostome means second mouth because the anus forms from the blastopore early in embryonic development. By contrast, protostome means first mouth because the mouth forms from or near the blastopore. Deuterostomia encompasses chordates, echinoderms, and hemichordates, while protostomia is almost every other bilaterian phylum. Arthropods, tardigrades, mollusks, brachiopods, nematodes, etc. With regard to those fossils, a species known as Ionanozoan, which is evidently synonymous with Hycuella, from 525 million years ago, looks overall superficially similar to the aforementioned stem vertebrates, but it lacks definitive diagnostic characters that would firmly place it as either stem vertebrates, or among other chordates, or other deuterostomes. First, Unanozoan has a segmented cuticle similar to Chinorhynchs, Loriciferans, and arthropods, all of whom are very distantly related to vertebrates. The developmental processes underlying segmentation in all bilaterians are guided by homologous genes, so the mere existence of segmentation doesn't lend any clues to which group this animal is in either. Unanozoan does have gill arches similar to vertebrates, however these arches are attached to sclerotized rods which extend anteroposteriorly that lack any homology in vertebrates. It is also difficult to tell if Unanozoan is coelomate or pseudocoelomate, so no help here. And a coiled gut and serially repetitive gonads occur across a wide range of bilaterians. Intriguingly, the gill arches are actually internal, opening to an atrium before reaching the outside, which is also the case in our deuterostome cousins, the lancelets and acorn worms. Overall, then, this suite of characteristics is a blur of features that doesn't permit us to confidently assign Unanozoan to either stem deuterostomes or stem protostomes, never mind stem vertebrates. Which is rather what we would expect early in a basal diversification at the root of the animal phyla we know today. Regardless of these fossils' affinities, a characteristic that is ancestral to vertebrates is gills. A long-standing hypothesis in zoology states that the gills of cyclostomes and gnathostomes evolved independently. After all, when you're small, you don't need an entire respiratory system for gas exchange. You can just exchange gases across the body wall. As stem vertebrates became larger, the need for a respiratory system increased, albeit at first, a small system that was a series of folds branching from the esophagus to the body wall. However, the gills of cyclostomes and gnathostomes form on different regions of the pharyngeal arches and arise from different epithelial tissues. Cyclostome gills arise exclusively from endodermal tissue. In bony fish, while the gills of sturgeons and lungfish arise from ectodermal tissues, the gills of zebrafish arise from endodermal tissue. To resolve what the ancestral state was, a team of researchers performed cell lineage tracing in the little skate, Leucorrhagia arenacea. Just like in cyclostomes, the skate's gills are endodermal in origin, implying a common endodermal origin for gnathostome and cyclostome gills. Another characteristic shared among all vertebrates is the camera eye. See the howler monkey's tail for a refresher on how phototransduction works. The camera eye is a type of eye shared by vertebrates and coleoid cephalopods, but since it arose independently in these clades, there are some obvious differences between the camera eyes of vertebrates and cephalopods. For one thing, the optic nerves of vertebrates pass in front of the retina, whereas they are behind the retina in cephalopods. Overall, though, they have most of the same parts. Importantly, they share a cornea, lens, vitreous humor, and retina. The cornea is a clear layer that protects the eye, the lens focuses light onto the retina, the vitreous humor is a clear fluid that fills the eye, and the retina collects information from light to pass to the brain. Using just these four characteristics, biologists Dan Eric Nilsson and Suzanne Pelger calculated how many generations it would take for a camera eye to evolve if each part changed just 1% in its dimensions or refractive index per generation. The pair found that the number of generations needed for such an eye to evolve is about 364,000 generations. Assuming the generation time of these organisms is a year, that would easily allow camera eyes to develop from just a patch of tissue covered with photoreceptors in one geological epoch. It makes sense then why camera eyes just appear fully formed at the base of the Cambrian, though in many forms in distinct lineages as the genetic mutations go their separate ways. We'll cover the actual evolution of the eye a few tales from now. Next, we meet our closest living relatives with whom we share a common ancestor that lived approximately 535 million years ago in the early Cambrian, the tunicates. Or perhaps even earlier if the baggy Ediacaran blobs described in a 2021 paper turned out to be tunicate ancestors. Tunicates are split by molecular genetics into two living clades, Appendicularia, also called Larvaceans, and Acidiacea, the sea squirts. Larvaceans are free-swimming throughout their entire lives, looking a bit like the ammocoete larvae of lampreys. Sea squirts, by contrast, look like Larvaceans, but only as juveniles. Almost all of the defining characteristics of chordates, pharyngeal arches, a dorsal hollow nerve cord, a notochord, and a postanal tail, are lost as the juvenile sea squirt metamorphoses. The sea squirt even reduces its cerebral ganglion during metamorphosis, which has given rise to the meme that sea squirts eat their own brain like an associate professor obtaining tenure. Adult sea squirts do, however, still have cerebral ganglia. Juvenile sea squirts are unable to eat, even though they do have a rudimentary digestive tract. Once enough development has occurred, the sea squirt settles headfirst onto sediment. It turns into a gelatinous bag with two siphons, the buccal siphon for water coming in, and the atrial siphon for water going out. The adult tunicate is surrounded by a covering called the tunic that is largely composed of a polysaccharide called tunicin, which is a type of cellulose. Inside the pharynx of the tunicate is the endostyle, one of those diagnostic chordate characteristics, that secretes mucus to capture food. In lampreys, the ammocoete has an endostyle that metamorphoses into the thyroid. In all more-derived vertebrates, including us, the endostyle is the thyroid. Remember from the axolotl's tale that metamorphosis in vertebrates is regulated by iodine-containing thyroid hormones, and this is true for tunicates too. Because tunicates are sister to vertebrates, there has been much debate regarding their evolution and its implications for the ancestors of vertebrates. Traditionally, tunicates have been considered as three clades: larvaceans, sea squirts, and another clade of free-swimming tunicates called salps, which includes all of Dapper Dinosaur's favorite animals. Previously, researchers thought that lancelets were more closely related to vertebrates than tunicates are, since lancelets are rather fishy overall. This led to the idea propounded by Walter Garstang, that biologist we met in the axolotl's tale, that tunicates and chordates generally were ancestrally sessile, as adults with motile larvae. After all, every animal that is sessile, as an adult, has motile larvae, whether sponges, corals, barnacles, etc. The ancestor of lancelets and vertebrates, then, was an individual who retained their motile juvenile characteristics into adulthood. That is, they underwent neoteny. According to genetics, however, tunicates nest closer to vertebrates than lancelets do, so this hypothesis has been discarded. Interestingly, Charles Darwin presciently argued in Descent of Man that tunicates are descended from ancestors with a motile adult stage. Darwin wrote, quote, "Ascidians are related to the vertebrata in their manner of development, in the relative position of the nervous system, and in possessing a structure closely like the chorda dorsalis of vertebrate animals. We should thus be justified in believing that at an extremely remote period a group of animals existed resembling in many respects the larvae of our present ascidians, which diverged into two great branches, the one retrograding in development and producing the present class of ascidians, the other rising to the crown and summit of the animal kingdom by giving birth to the vertebrata." Close quote. The clade that encompasses vertebrata plus tunicata is olfactores. This name refers to both clades possessing homologous olfactory apparatuses. The name was coined by paleontologist Richard P. S. Jeffries, who also proposed the calcichordate hypothesis. Briefly, this was the idea that a clade of Paleozoic deuterostomes called Stylophora were ancestral to chordates. Some stylophorans were asymmetrical, like early echinoderms, but others were more bilaterally symmetric. One feature of stylophorans is their single jointed appendage containing a tube. What exactly this appendage did has been disputed. One hypothesis is that it is an arm-like feeding appendage, and the tube is part of the hydrovascular system of echinoderms. But Jeffries flipped the animal, interpreting the structure as a tail with a possible notochord instead. That puts the stylophorans as stem chordates. Like echinoderms, stylophorans have a test composed of calcite plates, meaning that the ancestors of chordates had calcium plates too, hence calcichordate. However, researchers have recently been able to scan the internal fossilized soft tissue of stylophorans, and they found that the structure of the tube is most consistent with the echinoderm hydrovascular system and tube feet, securing the stylophoran position among echinoderms and therefore unrelated to the issue of chordate origins. But there's another twist to tunicate phylogenetics. Based on morphology, larvaceans are sister to all other tunicates, and salps are sister to sea squirts. Because two origins of an ancestrally free-swimming adult tunicate stage are less parsimonious than a single origin, this arrangement seemed to imply the common ancestor of all tunicates had a free-swimming adult. But genetics again threw a wrench into everything. While larvaceans are still considered sister to all other tunicates, salps actually nest within the sea squirts, meaning they obtained free-swimming adults independently of larvaceans. Therefore, we can't know based on extant tunicates alone what the adults of ancestral tunicates look like. Here, fossils come to the rescue. Shankuklava from China dates to 518 million years ago and is often considered the oldest stem tunicate. It possesses some, but not all, of the diagnostic tunicate characteristics like the eponymous tunic, pharyngeal basket, and buccal siphon. In 2023, a team of researchers discovered the 500 million year old megasiphon in Utah that does possess paired siphons and is broadly sea squirt-like in its morphology. Both Shankuklava and megasiphon are resolved as stem tunicates, indicating a sessile adult stage for ancestral tunicates. So to recap, tunicates are sister to vertebrates, switching with lancelets, stem tunicates developed a sessile adult, and larvaceans and salps independently return to motility in adulthood. On that note, we're pulling out of olfactores and uniting with the Lancelets at 540 million years ago, the very start of the Cambrian. The clade that unites the Lancelets with the tunicates and vertebrates is called chordata. The characteristics that are shared among these groups are pharyngeal arches, a postanal tail, a dorsal hollow nerve cord, an endostyle, chevron-shaped myomeres, also called V-shaped muscle blocks, and a notochord. We can also note important traits that vertebrates have that are either lacking or less complex in tunicates and lancelets. Recall from before that the endostyle is homologous to the thyroid gland, as is shown in lamprey metamorphosis. Although the dorsal nerve cord of lancelets and tunicate larvae is slightly enlarged, it's difficult to say whether they are true brains or not. Vertebrates clearly have a distinctive tripartite brain divided into a forebrain, midbrain, and hindbrain. Related to this is the lack of many sensory organs. Neither tunicates nor lancelets have an auditory or balance system that we see in our inner ear, nor do they have complex camera eyes, although they do have a simpler visual system. More on that later. We can also see a lack of a differentiated digestive system. Our pancreas is a specialized organ that contains exocrine cells for secreting digestive enzymes into the gut, and endocrine cells that secrete hormones like insulin into the blood to regulate blood sugar levels. In vertebrates, this system is separated from the main gut in various degrees depending on the group. But in lancelets and tunicates, cells that line their gut perform pancreatic functions, including secreting insulin-like proteins. This is similarly the case regarding the liver. The lancelet gut has a single, blind-ending pocket that branches off from the main gut, which is called the hepatic cecum. Again, it's not the same, but it performs many functions of a vertebrate liver. The kidneys are also simplified to the point where it's difficult to call them kidneys. In vertebrates, different versions of kidneys develop in a sequential manner, each more complex than the former. The pronephros, then the mesonephros, and finally the metanephros. Amniotes are the only ones that have the metanephros, which are the ordinary kidneys that you have. When the metanephros develop in amniotes like us, the pronephros and the mesonephros disappear, except for a few parts of the mesonephros that remain in the male reproductive system. In amphibians and fish, there is no metanephros. The mesonephros functions as the adult kidney, while the pronephros only functions in larvae. In jawless fish, the pronephros is the only kidneys that they have, even as adults. However, lancelets don't even have any nephrons with a Bowman's capsule. Instead, they use nephridia, wherein fluid with waste is excreted, which is wafted out by flagella of selenocytes. These paired nephridia ducts exit on the sides of the body and are tightly associated with the paired gill openings. Lastly is the circulatory system. As noted in the lamprey's tail, vertebrates have blood cells with independently exapted globin proteins that carry oxygen. However, tunicates and lancelets also do not have this. Their oxygen is dissolved in their blood plasma. They do have globin genes. In fact, many organisms, even bacteria, have globin genes, but these don't function in oxygen transport. They are involved in other functions, such as the metabolic defenses against oxygen and nitric oxides. They also don't have multi-chambered hearts. Tunicates do have again, it's difficult to call it a bona fide heart. It's a curved tube that pumps blood by peristaltic contraction. Also, a curious fact, the direction of blood circulation in tunicates periodically reverses. The few similarities with vertebrate hearts is the fact that they are powered by striated muscles and the structures are enclosed within a pericardium. On the other hand, lancelets have 4 main peristaltic vessels powered by smooth muscles and there is no pericardium. This fact that the, quote, hearts of our chordate relative consists of tubes and not chambers suggests that our hearts evolved from a tubular structure too. This is also what we see in our ontogeny, similar to how our inner ear bones develop from jaw-associated pharyngeal arches, or how flatfish first develop symmetrically before their faces become asymmetrical. We see that the embryonic precursors of the heart are tubular blood vessels that fuse and fold into themselves to construct the chambers. In the previous episode, we have noted that in vertebrates, the pharyngeal arches give rise to various structures, including the gills, parts of the throat, jaw, and face. The pharyngeal arches begin development as outpocketings of the endoderm that can be viewed from the outside as serial clefts. The arches are mostly filled with a type of tissue called neural crest, which is derived from ectoderm and will give rise to a variety of tissues, including neurons, melanocytes, cartilage, connective tissue, and smooth muscle tissue. Neural crest cells are a defining characteristic of olfactores, and without this tissue in the pharyngeal arches of lancelets, their arches are much simpler. As would be expected, the genes regulating the formation of pharyngeal arches are homologous across vertebrates. Perhaps more surprising is that the gills of hemichordates, like acorn worms, also express genes involved in chordate pharyngeal arches, like PAX1/9. This indicates that endodermal outpocketings giving rise to pharyngeal arches is more likely a basal deuterostome trait that was lost in echinoderms. This is confirmed by Cambrian stem echinoderms, like the stylophorans discussed earlier. While pharyngeal gills in most vertebrates function for respiration, in lancelets the function of the gills is merely for feeding. Lancelets stick themselves tail down in the sediment and filter particles out of the water. The endostyle gland that secretes mucus helps to trap particles in the pharynx, which is transported down to the guts, while water is free to flow out via the gills. They don't need to take in water for gas exchange because they're small enough to do that across the body wall. In other words, they breathe with their skin. Only as stem vertebrates took on a more active lifestyle and obtained larger body sizes, did the function of the gills switch from filtering water to breathing. The first pharyngeal arch was then later co-opted to form the jaw in stem gnathostomes, as we discussed in the lamprey's tail. As evolutionary history has gone on, the number of pharyngeal arches has decreased from 7 in jawless fish to ancestrally 5 in bony fish to just 3 in amniotes. In cartilaginous fish, the segmental pattern of the embryonic arches is seen in adults as the serial arrangement of gills. However, this is less obvious for ray-finned fish, who cover their gills with an operculum. The operculum is present in coelacanths and lungfish too, as well as some stem tetrapods like Panderichthes. More derived stem tetrapods like Tiktaalik and Acanthostega lost the operculum, with the posterior ends of the skull acting analogously, as we mentioned in the lungfish's tail. Along the stem lineage of tetrapods, the 2 posterior pharyngeal arches ceased forming gill buds since respiratory functions shifted to the lungs. This resulted in a decrease from 5 to 3 arches. One issue that was caused by the transition to land was that fish readily take up calcium from the surrounding water. Obviously, this can't be done by land-living tetrapods, so new mechanisms for regulating calcium homeostasis had to be put into place. This is done by the parathyroid glands. There are 4 of these in humans located in our throat on the thyroid gland, which, remember, is an organ that dates to the very beginning of chordates. The parathyroids sense changes in the body's calcium levels, and if the levels are too low, the parathyroid glands release parathyroid hormone that liberates calcium from such places as bone. The parathyroids arise embryonically from pharyngeal pouch endoderm, and fascinatingly, the gene that regulates parathyroid development is a gill bud development gene called GCM2. Further, homologs of parathyroid hormone genes are expressed in fish gills, and fish gills express homologs of the calcium receptor that the parathyroids use. All of this is to say that we humans and rabbits and elephants and owls and geckos still have gills. They're just the parathyroids now. Next, let's look at the notochord and dorsal hollow nerve cord. The notochord is the character for which chordates are named. It's an elastic, rod-like organ made of glycoproteins and collagen that resembles cartilage, but it's not quite the same. In vertebrates beside the hagfish, the function of the notochord is superseded by a cartilaginous or bony vertebral column, while remnants of the notochord become part of the intervertebral discs. Sessile tunicates completely lose the notochord. For lancelets and motile tunicates, the notochord retains its embryonic structure throughout the animal's entire life. In mammals, the notochord itself develops from endodermal tissue very early in embryo, just 17 or so days after fertilization. Because it runs along the midline of the body from head to tail, that makes the notochord extremely useful for organizing tissues. Proteins secreted by the notochord participate in the differentiation of nerve cells that form the spinal cord, as well as the differentiation of connective tissues and muscles that form down both sides of the body. Another function of the notochord is for swimming. For small body sizes, the notochord provides enough rigidity that allows lancelets and motile tunicates to swim by lateral or side-to-side undulation by contracting their V-shaped muscle blocks. As one paper puts it, quote, "The most typical hydrostatic skeleton among invertebrates results from the constraint of internal fluids and soft tissues by a rigid or elastic body wall. A second kind of hydrostatic skeleton is an internal rod, the notochord, which functions as a flexible compression strut," close quote. As only chordates possess the notochord, we must ask, is the notochord a novel characteristic that originated in chordates, or was the notochord exapted from a previously existing structure? The notochord was first discovered in chicken embryos by embryologist Carl Ernst von Baer. This is the guy who famously argued against the idea that ontogeny recapitulates phylogeny, or the idea that, as we develop, we pass through earlier stages of our evolutionary history. Technically, the modern version of this idea is a bit more nuanced, but that's a conversation for another time. Von Baer instead argued that different lineages diverged from different embryos. Indeed, this is what we see with the vertebrate phylotypic stage, that embryonic stage famously, or perhaps infamously, represented by Ernst Haeckel's non-fraudulent drawings. Von Baer argued that the notochord was one of the identifying characteristics of vertebrates, identifying vertebrates as one of Georges Cuvier's 4 embranchments. These were the divinely created kinds. Cuvier pinned cnidarians and echinoderms, the radially symmetrical animals, as the second embranchment, annelids and arthropods, the segmented animals, as the third, and mollusks, the fourth. Von Baer and Cuvier argued that evolution could never bridge the gap between embranchments, but of course, by allowing all vertebrates to be one kind, that means lampreys and elephants share a common ancestor. That's quite a lot of evolution. At any rate, from 1863 to 1864, a Russian student, Alexander Kovalevsky, from the same department as von Baer at St. Petersburg University, formally challenged von Baer's embranchment assertion after publishing a series of works on the development of lancelets and tunicate larvae. Von Baer had pinned tunicates as mollusks based on incorrect homology between tunicate and bivalve ciliated gill slits, but as Kovalevsky showed, tunicate development was a dead giveaway for vertebrate cousinship. Cuvier and von Baer's embranchments had been violated. From there, other researchers tried identifying homologies between the notochord and invertebrate organs, but with little success. A large part of the reason for this is that the phyla that researchers tried to unite with vertebrates turned out to be very distantly related to them, like arthropods, annelids, nemerteans, phoronids, and even cnidarians. Other homolog attempts involved invertebrates known to be more closely related to vertebrates, the hemichordates and echinoderms. Hemichordates have a hollow tube that extends from the pharynx to the proboscis called the stomochord, and echinoderms have their water-filled vascular system. The latter appears quite unlikely to be related to the notochord, as the notochord never passes through a coelom-like stage of development. As for the stomochord, it could possibly be related to the notochord, since both are the source of hedgehog signaling. However, none of the other genes upstream or downstream of notochord-forming genes could be detected in the stomochord. Currently, the best hypothesis is that the notochord evolved from some mid-dorsal cells that acquired signaling functions, which had previously been occupied by the endoderm and ectoderm. Obviously, much more is left to learn about the notochord, and that will come from understanding more deeply the genetics and development of the invertebrate chordates. Well, can fossils tell us anything about the origin of the notochord? Sort of. We need to meet the well-known Burgess Shale fossil Pikaia, which was made famous by Stephen Jay Gould's 1989 book Wonderful Life. Gould's work was a celebration of the discovery of the bizarre menagerie of Middle Cambrian fossils from British Columbia. Though Gould's ideas about the Ediacaran fauna being totally unrelated to the later Cambrian ones, or the Cambrian fauna being more morphologically diverse than modern fauna, are no longer considered valid, the book is still important for the public intrigue it generated around Cambrian animals. In fact, Wonderful Life ends with a discussion of Pikaia. I think the final paragraph is beautiful, and I can't help but quote it here. Quote, "And so, if you wish to ask the question of the ages, why do humans exist, a major part of the answer, touching those aspects of the issue that science can treat at all, must be, because Pikaia survived the Burgess decimation. This response does not cite a single law of nature. It embodies no statement about predictable evolutionary pathways, no calculation of probabilities based on general rules of anatomy or ecology. The survival of Pikaia was a contingency of just history. I do not think that any higher answer can be given, and I cannot imagine that any resolution could be more fascinating. We are the offspring of history and must establish our own paths in this most diverse and interesting of conceivable universes, one indifferent to our suffering, and therefore offering us maximal freedom to thrive or to fail in our own chosen way." Close quote. 2-inch long, 508-million-year-old Pikaia was first described by Charles Doolittle Walcott in 1911 as a polychaete worm based on the presence of repeating segments, but an extensive reanalysis of Cambrian animals by paleontologists Simon Conway Morris and Harry B. Whittington in the 1970s reached a different conclusion. Pikaia bears a series of vertical striations resembling somite boundaries, which implies the existence of serial muscle blocks called myotomes, and Pikaia has a flattened, streamlined body for undulatory motion. These characteristics are similar to those seen in modern Lancelets, earning Pikaia its place among the chordates. It even has a structure running down its dorsum that might potentially be a notochord. Pikaia does, though, have some distinctly unchordate-like characteristics, such as its bilobed head and 9 paired filamentous appendages. Despite this, it has the notochord, so it's a chordate, right? For one thing, the dorsal organ of Pikaia isn't a rod like the Lancelet's notochord. It's an elongate ovoid. Assuming the typical orientation for Pikaia and that the dorsal organ is indeed dorsal, we don't know if it is actually the notochord, whether it sits above or below the dorsal hollow nerve cord, or is the dorsal hollow nerve cord itself. If the dorsal organ is the notochord, then it could be filled with turgid vacuolate cells, similar to the situation in extant notochords, and, interestingly, the hemichordate stomochord. That the dorsal organ is a notochord and a site of attachment for the myotomes is the least radical of the potential options, which would ensure Pikaia's place as a chordate or stem chordate. Cathamyrus is another early chordate from the Mao-Chien-Shen shales, dating to 518 million years ago, slightly older than Pikaia. Like Pikaia, Cathamyrus has myotomes and a potential notochord, but it also preserves evidence of pharyngeal gills. Now we turn to the dorsal hollow nerve cord, or neural tube. After the notochord forms embryonically, it induces the ectoderm above it to invaginate until a tube is formed. This tube, or cord, is the precursor to the brain and spinal cord, the central nervous system. Where did the neural tube come from? The tissue patterning of the neuroectoderm that gives rise to the neural tube corresponds to similar domains in the whole body axis of protostomes, indicating that such patterning is ancestral to the protostome-deuterostome split. However, possessing a hollow nerve cord is a chordate invention, as acorn worms have both dorsal and ventral nerve cords. By contrast, protostomes have a ventral nerve cord, which has contributed in part to the development of the dorsoventral inversion hypothesis for the origin of chordates. The inversion hypothesis essentially states that the bodies of deuterostomes are flipped relative to the bodies of protostomes. Aside from the nerve cord of protostomes versus chordates, certain morphogens like BMP and chordin are flipped in their expression between these two clades. How could this have possibly happened? Understand that inverting all of one's organs can happen, and when it does, it doesn't normally have harmful side effects. This condition is called situs inversus, and it affects about 1 in every 10,000 people. Likewise, the original inversion of a protochordate probably didn't have any adverse side effects, otherwise we wouldn't be here today. Exactly how this inversion happened is still discussed. There is a hypothesis that puts a twist on this inversion, quite literally. Not only is our nervous system upside down relative to protostomes like insects, it's also contralateral. In general, the left side of our brain controls the right side of our body and vice versa. This is due to the fact that major nerves cross one another within the central nervous system behind the forebrain, which are called decussations. In the peripheral nervous system, these crossing points are called chiasmas, one of which is the optic chiasm. This anatomical quirk has been puzzling for a long time. The axial twist hypothesis proposes that the anterior portion of the head of the ancestors of all vertebrates, and perhaps all chordates, twisted 180 degrees relative to the rest of the body. This twist is also observed during the embryonic development of zebrafish. They also propose that the reason for this might have been similar to what happened to flatfish that we discussed before. The ancestor lived on the seafloor, with one eye looking into the ground. To compensate, the face turned 90 degrees. However, when they later abandoned the seafloor and began swimming upright, the face had to turn again, but it overcompensated by another 90-degree twist. Related, the last characteristic of chordates we need to discuss is, and this might surprise you, the mouth. As stated earlier, the dorsoventral inversion occurred in stem chordates. However, the side of the body where the mouth is located is basically what defines the ventral side. So, tautologically, both protostomes and deuterostomes have their mouth on the ventral side. This would pose a problem for a simple inversion scenario where the whole body just flipped. That would have simply put our mouth on the same side as the nervous system, just as it is in protostomes. But the aforementioned axial twist hypothesis accounts for this, where the front part of the body, including the mouth, inverted relative to the rest of the body. So while the mouth of protostomes and non-chordate deuterostomes develops on the original ventral side, the mouth of chordates moved towards the original dorsal side, which is our ventral side. But there is yet another thing going on with our mouths. In 2016, Kaiji et al. Showed that in tunicates and vertebrates, the mouth develops from ectoderm that gives rise to a number of placodes, but the lancelet mouth develops from a somite on the left side of the head. The lancelet's mouth then moves to the anterior of the head during development. This could imply that the mouths of lancelets, and perhaps ours as well, used to be a gill opening. However, the researchers suggest that the mouths of vertebrates and tunicates is a novelty independent from that of the lancelet, and that their original mouths in both were lost. The same researchers who proposed this also suggest a homology between the lancelet's mouth and the madreporite of echinoderms, or the proboscis pore of hemichordates. Echinoderms and hemichordates are the closest relatives of chordates. Hemichordates also have gills, but some of their gill openings appear to have lost their connection to the pharynx and some also have lost connection with the outside. While modern echinoderms have no gills whatsoever, some of their fossil relatives did have gills. These pores in echinoderms and hemichordates only extend into the body cavity and are used to control various appendages. Among the hemichordates, the terebranchs specifically have these branched appendages used to filter food, while both terebranchs and acorn worms have a cavity to control their acorn-shaped proboscis at the front end. The latter connects to the outside via the proboscis pore, the homologue to the lancelet's mouth. In echinoderms, the homologue is the madreporite, which also leads into body cavities. In some echinoderms, like sea stars, this has been elaborated into an extensive hydrovascular system with canals that control thousands of tube feet. So with all of that now covered, we can now meet the lancelets properly. Lancelets are encompassed by the subphylum Cephalochordata, and they look a bit like tiny finless fish. They're small, laterally compressed animals that filter feed particles drifting through the water using their oral tentacles. Unlike vertebrates and tunicates, they retain their pharyngeal slits, notochord, dorsal hollow nerve cord, postanal tail, and endostyle throughout their life. Lancelets also have a single frontal cup eye instead of our paired eyes, but they supplement light input to the frontal eye with input from other light-sensitive structures called Joseph cells, the organ of Hesse, and the lamellar body, all of which are in the dorsal nerve cord. By comparing modern lancelets with fossils like Hycoichthyes, we can infer that the ancestor of chordates may have looked a bit like a lancelet. However, it certainly would not have itself been a lancelet. This is the moral of the lancelet's tale. Lancelets are just as modern as we are, so neither lancelets nor any other modern organism should be thought of as primitive. They have participated in the last 540 million years of evolutionary history, just like us. This is also the reason why the argument, if humans are descended from monkeys, then why are there still monkeys, doesn't make any sense. While we can't say any modern organisms are our direct ancestors, we typically can't say this for fossils either. Given the prodigious number of extinct individuals and species, the odds that we will find a fossil that is either a member of a species directly ancestral to us or a particular individual that is directly ancestral to us is, given enough time, vanishingly small. There are a few very recent, as in the last few hundreds of thousands of years, species that we can point to as being directly ancestral, like Homo ergaster. The best we can say is that, to a reasonable approximation, some modern or fossil organisms are similar in certain ways to our ancestors. So that's the lancelet's tale. Lancelets are our most distant extant chordate cousins, and we share several important characteristics with them. Despite this, they are not our direct ancestors. We both share a common ancestor who lived in the early Cambrian that was neither a lancelet nor a vertebrate. So, thanks for watching, and we'll see you all next time.